In this video, we're going to continue with patterns of inheritance and continue with multiple allele inheritance. So far we've seen two alleles that will give the trait a dominant, a recessive. However, sometimes we need more than the two alleles to express the trait. And an example of that, this multiple allele inheritance, is the ABO blood groups. Right? Someone can be type A blood, B blood, AB blood, or O blood, and that actually comes from three alleles. So the genes, or the alleles, are I with an A for the A antigen, I with a B for the B antigen, or just the I means they don't have A or B antigen, which we call O blood type there. So A and B are codominant. Both are expressed if someone has both of these alleles. I is a recessive type for the O blood type. Remember, O just really means 0. That means this person's red blood cell doesn't have the A antigen or the B antigen. So if we look at our possibilities here, if someone has both recessive blood type genes for the ABO system, small I, right there, I guess that should have been a small I, they're type O. They don't have any antigens of the ABO system. Well, at the other extreme, if one parent gives the offspring the A antigen allele, and the other parent gives the offspring the B antigen allele, well, both of those are expressed. Therefore, some of their red blood cells have A antigens, some of their red blood cells have B antigens. Therefore, this person is AB blood type because they have both types of antigens on their red blood cells. And how is a person type A blood? Well, that means both parents gave this A allele, or one parent gave the A allele and one parent gave the recessive, which is O. And remember, O means nothing. And if there's nothing, well, there's nothing to express, but this person is expressing the A, so they'll be type A. And the same scenario with type B. Either they got B's from both their parents or from one of their parents where the other parent had the recessive O. Yet another type of inheritance is sex-linked inheritance. These are traits that are inherited on the sex chromosomes, the X and the Y. Now, there's 23 pairs of homologous chromosomes. However, the pair, that's the X and the Y, the sex chromosomes, aren't really homologous to each other. There's a couple homologous genes that on each of them, but those chromosomes are actually very different from each other. The X chromosome bears 1,400 genes, where the Y chromosome only carries about a small fraction of that, 200 genes. And typically, it's this Y chromosome is just coding for male traits, which makes sense because females don't have a Y chromosome. Right? Females will have 2 of these X ones that'll work as homologous pairs, right? But males will have 1 X and 1 Y, meaning whatever's on the X, it's probably going to get expressed because it doesn't have a homologous chromosome to override it with a dominant gene. And because of these differences between the X and the Y, not a whole lot of crossing over going on because there's very few regions that can participate in crossing over because very few regions are homologous to each other. So genes that are found only on the X chromosome, which there's quite a few of them, are called X-linked genes, sometimes called sex-linked genes or just X-linked genes. So let's look over sex-linked inheritance using the example of color blindness because normal color vision is found on the X chromosome. So here is a male with normal vision, large N. So when they produce, this male produces its gametes or sperm, half the sperm will have that X with normal vision, half their sperm will be a Y that doesn't have a gene for vision. When I say vision, normal color vision versus color blindness. Here's a female who has an X with with normal color vision and an X with color blindness. This female will not be colorblind because that large N is dominant over the small n. So normal color vision is dominant over color blindness, therefore the phenotype is normal color vision. But when this female produces her gametes, or egg cells, half will have normal color vision. The other half of the eggs will have color blindness gene associated with it. So let's cross these two individuals, and of course, half are female, half are male. That's always the case here as far as probability goes. But we see that, let's go through the female genotypes and phenotypes first. This female has normal vision because they're homozygous dominant for normal color vision. This female will have normal color vision because they're heterozygous for heterozygous, meaning this one dominant normal color vision is dominant over the recessive gene for color blindness. So in this case, females will all have normal color vision. On this side of the Punnett square are the male offspring, and we can see this male offspring has normal color vision because of the dominant gene, where this male offspring will be colorblind because they have that small n. There is no large n on the Y chromosome because the Y doesn't produce that gene. It doesn't exist on the Y chromosome. So in an X-linked gene, the male offspring only needs to have one recessive gene to express that trait because there's no homologous chromosome to override it. So there is no heterozygous possibility in the male. So this means more than half, 3 out of 4, 75% would have normal color vision, but 25% are colorblind, and that will always be the male in this case based on our Punnett square. The only way a female could become colorblind if they receive the small n from both parents, which is not the case here because this male has normal color vision. So a good summary is right here. Females must have recessive alleles on both X chromosomes to express. Another type of inheritance, of genetic inheritance, is polygene inheritance. Traits that are a result of actions of several gene pairs at different locations. So, the more genes are involved in a trait, the more phenotypic variation will be seen. And we see this a lot in humans. The results are continuous phenotypic variation between the two extremes. For example, in humans, we don't have a tall gene and a short gene, meaning you're either 6'5" or 5'1". We have everything in between, and even a larger range than that, because it's a result of many genes put together. Right? That's polygenic inheritance. It's not one or the other, it's the whole range, this continuous phenotypic variation in between the two extremes. Examples would be skin color, height, my example, intelligence, metabolic rate. So let's look at the skin color example now with the figure on the next slide. So here we're looking at polygenic inheritance for skin color, alleles for dark skin, A, B, and C. There's 3 different ones. They're dominant, but incompletely dominant over light skin. Here, we're going to, well, actually, let's go through this before we read here. So here's 2 parents. This person has all 3 recessive genes, homozygous recessive for very light. So this is as light a skin color as possible. Where at the other extreme, we have very dark, meaning homozygous dominant for all 3 genes for dark color skin. So if these 2 parents mate, all the offspring will be heterozygous for all 3 genes that have to do with skin color. So we can see this genotype is identical to this genotype, which is right in between the lightest and the darkest here which is the center of this graph. But what if we take a person who is heterozygous for all three, cross them with a person, right, sexual reproduction, who is also heterozygous for all three? Well, now this polygenic inheritance will give us all of these different possibilities, and we can see that 1 out of 64 will be very light, just like one of the grandparents. 1 out of the 64 will be very dark, like one of the grandparents. And then we have all of these other possibilities in between. 6 out of 64, second from each end, 15 out of 54, third from each end, and right in the middle, the most with the most possibility is 20 out of 64, and that gives us this bell-shaped curve here. So this is polygenic inheritance, and our example with skin color actually uses 3 genes instead of 1, where offspring of 2 heterozygotes, there's the heterozygotes, the offspring of 2 of them, would have this wide variation of possible pigmentations. And if we chart it like we did here, we get the bell-shaped curve. And a good place to end this video here is that there's also environmental factors that affect gene expression. And environmental factors will influence it after birth. So someone has a specific genotype which would predict a specific phenotype. However, some environmental factor is not making that trait or expression or phenotype what we would expect it to be. Like poor nutrition can affect brain growth, body development, height. Right? Someone has the genes to be a specific height. That person might not get there with poor nutrition, for example. So that's an environmental factor. Hormonal deficits in children can lead to abnormal skeletal growth and proportions. So there is an environmental factor to gene expression where what the expected phenotype might not get there because of examples like poor nutrition.